Welcome to our lecture on animal metabolism, nutrition, and digestion. We've all likely heard the phrase, "you are what you eat." Not only does this imply that animals build their bodies and obtain energy from the food they eat, but it also includes that an animal's very appearance often reflects what they eat. In this lecture, we'll discuss four main themes. The first is how metabolic rate depends on activity level, body size, and body temperature. Then, we'll discuss how an animal's diet supplies the energy the animal needs for homeostasis and the essential nutrients it cannot synthesize on its own. We'll move on to talk about the fact that animals have different adaptations for feeding, and then finally, that the mammalian digestive tract has regions specialized for different functions, including digestion, absorption, storage, and elimination. Let's begin by delving into a discussion of animal metabolism. The biochemical pathways that make up animal metabolism are remarkably conserved among diverse groups of organisms because these pathways evolved early in the history of life. Nearly all animals depend on three main molecules as sources of energy and building blocks for growth and development. These are carbohydrates, fats, and proteins. These molecules are broken down, and in the process, the energy in their chemical bonds is transferred to biologically useful forms, typically adenosine triphosphate, or ATP. The breakdown of carbohydrates, fats, and proteins to produce ATP involves a linked set of chemical reactions in the process of cellular respiration. Reactions that break down food sources to fuel the energy needs of a cell are referred to as catabolic. Reactions that result in net energy storage within cells in the organism are anabolic. The details of cellular respiration are covered in another term of this introductory biology series, but let's discuss the process in general here. First, glucose can be partially broken down in the absence of oxygen by glycolysis. This process occurs in the cytosol of the cell, and it results in 2 molecules of pyruvate and 2 molecules of ATP. The next step depends on whether or not oxygen is present. If oxygen is not present, or is present only in small amounts, pyruvate is converted by fermentation into lactic acid during anaerobic metabolism. The physiological advantage of anaerobic metabolism is that it provides rapid but short-term energy to the cell and the organism. On the other hand, aerobic respiration requires oxygen. This process is carried out within the mitochondria of eukaryotic animals and provides a steady supply of ATP for longer-term sustainable activity. In this process, pyruvate is processed by the citric acid cycle rather than converted to lactic acid. Next, the electron transport chain and oxidative phosphorylation result in more ATP production. Along with ATP production, these steps consume oxygen and produce water. Overall, aerobic respiration results in the formation of a total of 32 molecules of ATP when 1 molecule of glucose is broken down, much more than the 2 molecules of ATP from 1 molecule of glucose produced anaerobically. Lipids are another important energy source for most animals. Lipids consumed in the diet, such as triacylglycerol, are broken down to glycerol and free fatty acids. These components enter glycolysis, or the citric acid cycle, to yield ATP by mitochondrial electron transport. Animals rely on fats for long-term energy storage. Proteins consumed in the diet are another useful energy source for animals. Proteins, which are needed for building and maintaining the body, constitute the enzymes and structural elements of cells and tissues. Only following prolonged food deprivation, when fat and carbohydrate reserves are depleted, do animals break down protein reserves to form ATP. An animal's overall rate of energy use defines its metabolic rate. Metabolic rate can be measured by the animal's rate of oxygen consumption, which in turn reflects the aerobic production of ATP. Metabolic rate is affected by many factors, one of which is the activity level of an organism. When an animal shifts from rest to activity, its metabolic rate and oxygen consumption rise to meet its increased demand for ATP. The onset of activity requires immediate energy, which in animals is provided by specialized energy stores in their tissues. Although glycolysis produces relatively few ATP molecules per molecule of glucose broken down, the reactions are extremely fast, providing a rapid, short-term supply of energy for animals. Animals rely on anaerobic glycolysis for short bursts of intense activity. With longer activity, the ATP needs of the animal are met by aerobic respiration in mitochondria. The rate of oxygen consumption initially increases, then levels off. At this point, the animal's need for energy is being met entirely by aerobic respiration. Once mitochondrial ATP production increases, the steady-state production of ATP depends on the adequate transport of oxygen to the tissue by the cardiovascular system, as well as by diffusion of oxygen into mitochondria. When activities end, the animal's oxygen consumption rate declines but does not immediately return to resting levels. The elevated consumption of oxygen following activity is the animal's recovery metabolism. It represents the continued metabolic energy required to reestablish the resting metabolic state of the cells. During recovery metabolism, cells resynthesize depleted ATP stores and metabolize the end products of fermentation, particularly lactic acid. In addition to activity level, an animal's size influences its metabolic rate. At rest, large animals consume more energy and have higher metabolic rates than smaller ones. This figure shows how resting, or basal, metabolic rate does not increase linearly with an animal's mass. Instead, measurements of resting metabolic rate in a wide range of organisms shows that metabolic rate increases with animal mass raised to the 3/4 power. As a consequence, the average rate at which each gram of body tissue consumes energy is less in larger mammals compared to smaller ones. Recall that larger organisms also have lower surface area per volume of body tissue. This scaling pattern of cellular energy metabolism is remarkable because it holds across a diverse size range of unicellular and multicellular organisms. Metabolic rate is also affected by an animal's internal body temperature. Temperature affects the rate of chemical reactions, which in turn determines how fast fuel molecules can be mobilized and broken down to supply energy for a cell. Animals that produce most of their own heat as byproducts of metabolic reactions, including the breakdown of food, are endotherms. These animals usually maintain a constant body temperature that is higher than that of their environment. The control of core body temperature, or thermoregulation, is a form of homeostasis and requires the coordinated activities of the nervous, muscular, endocrine, circulatory, and digestive systems. Mammals, including humans, and birds are examples of endotherms. This maintenance of body temperature means that endotherms typically have high metabolic rates that support sustained behavioral activity over a broad range of external temperatures. Animals that obtain most of their heat from the environment are ectotherms. Ectotherms often regulate their body temperatures by behavioral means, such as moving into or out of the sun. Most fish, amphibians, reptiles, and invertebrates are ectotherms. The activity level and metabolic rate of ectotherms both increase with increasing body temperature. However, ectotherms have metabolic rates that are about 25% of endotherms of similar body mass and at similar body temperatures. Although they can achieve activity levels similar to those of endotherms when their body temperatures are similar, ectotherms cannot sustain prolonged activity. Endotherms are sometimes referred to as warm-blooded and ectotherms as cold-blooded, but these terms are misleading because, depending on the environmental conditions, ectothermic animals can have core body temperatures higher than endothermic animals. Like core body temperature, blood glucose levels, and blood pressure, the energy balance of an organism is often maintained at a constant level. An animal in energy balance takes in the same amount of calories of energy from food as the energy that the animal expends. Energy is used to do work, things like maintaining tissues, growing, moving about, and the like. Energy required for basic life processes account for the majority, about 70%, of energy use. The remaining 30% can be used for physical activity, with higher levels of activity using more energy. When energy intake does not equal energy used, there is an energy imbalance. If an animal eats more food than it requires, energy stores such as fat deposits grow over time. During prolonged periods of an inadequate food supply or starvation, an animal consumes its own internal fuel reserves. Starvation forces animals to deplete their glycogen and fat reserves first. Then, if no food is found, they resort to protein stores, primarily in muscle tissue. Excessive intake of food calories has led to an increased and now critical public health problem of obesity. Obesity is now an epidemic in many industrialized nations, including the United States, where about 36% of the adult population is considered obese. It is a major public health concern because it increases the risk of diabetes, heart disease, and stroke, and contributes to a shorter lifespan. Animals' metabolic pathways enable them to obtain energy from the environment as well as to synthesize many of the compounds needed to sustain life. However, some nutrients necessary for life cannot be synthesized by an animal's metabolism. Therefore, these nutrients must be acquired in the food that they eat. An essential amino acid is an amino acid that cannot be synthesized by cellular biochemical pathways and instead must be ingested. Most animals can synthesize about half of their amino acids. Humans are unable to synthesize 8 of the 20 amino acids, and we have to obtain these 8 essential amino acids in our diets. Dogs and cats must obtain even more amino acids from their diets. Essential minerals, shown here in red, are chemical elements other than carbon, hydrogen, oxygen, and nitrogen that are required in the diet and must be obtained in the food that an animal eats. These include such elements as calcium, iron, phosphorus, potassium, zinc, and magnesium. Calcium is required for building skeleton, and iron in hemoglobin binds oxygen and transports it in the blood. Humans typically obtain the minerals sodium and chloride, which ionize to form salt crystals, through common table salt and other foods. Many wild animals seek exposed rock that they lick to obtain minerals and salts. Vitamins are organic molecules that are required in very small amounts in the diet. Vitamins have diverse roles, with some binding to and increasing the activity of particular enzymes, and others that act as antioxidants or chemical signals. The 13 vitamins listed here have been identified as essential for humans. In humans, as well as in other animals, vitamin deficiency can have serious consequences. For example, humans and other primates cannot synthesize vitamin C. If they do not ingest sufficient vitamin C, humans develop scurvy, a disease characterized by bleeding gums, loss of teeth, and slow wound healing. Deficiencies of vitamin B1, B6, and B12 can cause nervous system disorders and various forms of anemia. Vitamin D is essential for the absorption of calcium in the diet and thus to skeletal growth and health. Animals have evolved a variety of adaptations to enhance their ability to acquire energy and nutrients from the environment. Animals eat other organisms, whether they are other animals, plants, or fungi. Herbivores are adapted to eat plants, carnivores are adapted to eat animals, and omnivores are adapted to eat both. Regardless of food source, animals have evolved a diversity of ways to capture food and mechanisms for breaking that food before it's digested in the gut. The most common form of food capture by animals is suspension filter feeding. This form essentially involves passing water with food suspended in it through a sieve-like structure. Since water is key to this process, filter feeding is possible only in aquatic environments. This process evolved multiple times independently on many different branches of the tree of life, so it represents a form of convergent evolution. Many worms and bivalve mollusks, such as scallops, shown here, clams, and oysters, pump water over their gills to trap food particles suspended in the water. Other aquatic organisms, such as baleen whales, move water with food particles suspended in it through filters in their oral cavity and then convey the food into the gut. On a side note, notice those black dots that are on either fringe of the scallop's shells? These are actually photoreceptive rudimentary eyes, another example of convergent evolution. Many large aquatic animals apprehend prey by suction feeding, where a rapid expansion of the fish's mouth cavity draws water and the desired prey into the mouth. After the fish closes its mouth, the water is pumped out of the mouth cavity past the gills. The prey is trapped inside the mouth, moves into the pharynx, part of the throat, and is broken up by special pharyngeal jaws, a second set of jaws in the throat, before the prey is swallowed. Suction feeding allows organisms to be sit-and-wait predators, hiding within a coral reef or under a rock before rapidly striking to capture prey moving in front of them. Many insects that bite to obtain a blood meal also rely on suction to draw the blood of their prey into their digestive system. Young mammals feed by suckling milk from their mother's breast, using their tongue and also generating suction as fish do. Larger fish and marine mammals like sharks, whales, and dolphins actively swim to capture their prey. Their larger size and speed enables them to catch smaller or similar-sized prey. The evolution of jaws and teeth considerably enhanced the ability of these and other vertebrate animals to capture great amounts of food, allowing them to lead a more active and energetically demanding lifestyle. Jaws and teeth were an important evolutionary innovation for active predators and are key to the evolutionary success of vertebrates. Jawed fish evolved from jawless ancestors, and jaws can be found in present-day fish, amphibians, reptiles, and mammals. Jaws evolved from cartilage that supports the gills, providing a spectacular example of an organ adapted for one function, gill support, changing over time to become adapted for an entirely different function, predation. Some of the first vertebrates, the jawed fishes, became dominant in their aquatic environment through the ability to swim and bite forcefully to obtain their food. Among vertebrates, mammals evolved a specialized jaw joint, the temporomandibular joint, as well as a great diversity of specialized forms of teeth. These teeth have cutting and crushing surfaces, enabling mammals to break down a variety of foods mechanically before swallowing them. Teeth in the front of the mouth, the incisors, are specialized for biting. Other teeth, such as the canines of dogs, cats, and other carnivores, are specialized for piercing the body of their prey. Premolars and molars are teeth in the back of the mouth that are well adapted for crushing and shredding tougher foods, such as meat and fibrous plant materials. Arthropods, such as insects and crustaceans, lack jaws and teeth, yet they also capture, manipulate, and break down food. To do this, some used paired mouthparts called mandibles. Mandibles are located outside and in front of the mouth and are used to capture and physically break down food before it enters the mouth and then the gut. Mandibles have evolved a great diversity of structures and are well adapted to different sources of food. After animals obtain food, they break it down through the process of digestion. The food is isolated in a specialized organelle or compartment so that it can be broken down chemically without damage to other cellular organelles or structures of the body. Most animals use the process of extracellular digestion, in which food is isolated and broken down outside a cell in a body compartment. Following digestion, the breakdown products are taken up into the bloodstream in a process called absorption. Many animals have digestive systems that allow the transport of food by a digestive tube that runs from an animal's mouth to its anus and is known as the gut or digestive tract. Because the food is moved in a single direction, particular regions of the digestive tract can be specialized for different functions, including the mechanical and chemical breakdown of food, absorption of released nutrients, and storage and elimination of waste products. The digestive tract of animals are commonly divided into a foregut, midgut, and hindgut. The foregut includes the mouth, esophagus, and stomach or crop, which serves as an initial storage and digestive chamber. Next is the midgut, which includes the small intestine, where the remainder of digestion and most nutrient absorption takes place. Finally, the digested material reaches the hindgut, which includes the large intestine and rectum. Water and inorganic molecules are absorbed, leaving the waste products or feces, which are stored in the rectum until being eliminated from the body. Food and its breakdown products move through the gastrointestinal tract by peristalsis, waves of smooth muscle contraction and relaxation. Macromolecule digestion takes place in various sections of the digestive tract. The digestion of carbohydrates starts in the mouth by secretion of the enzyme salivary amylase. Carb digestion continues in the small intestine with the aid of pancreatic amylase and lactase enzymes. The digestion of protein begins in the stomach by the enzyme pepsin and continues in the small intestine by trypsin. The digestion of lipids begins in the mouth through breakdown by lingual lipase, continues in the stomach through gastric lipase, and ends in the small intestine through pancreatic lipase. Most animals jumpstart digestion by breaking down food mechanically, which increases the surface area exposed to taste buds in the tongue and available for enzyme action. In mammals, food entering the mouth is mixed with salivary secretions that contain amylase, an enzyme that breaks down carbohydrates, to begin the digestion of sugars and starches. Mammals and other land vertebrates have a muscular tongue that facilitates food manipulation and transport within the mouth cavity. Swallowing, which is controlled by the autonomic nervous system, is a complex set of motor reflexes carried out by several muscles and structures in the rear of the mouth and in the pharynx. The pharynx is the region of the throat that connects the nasal and mouth cavities. Once initiated, swallowing reflexes occur without voluntary control. During swallowing, food is pushed through the pharynx, over the epiglottis, a flap of tissue that protects food from entering the trachea and lungs, and into the esophagus, a tube that connects the pharynx to the stomach. Birds, alligators, crocodiles, and earthworms break down food into smaller pieces further along their digestive tracts in the gizzard, a compartment with thick, muscular walls. After being ingested and mechanically broken down, the food is transported into a stomach, one of the main sites of protein and fat breakdown. Hydrochloric acid and digestive enzymes break down protein and fat macromolecules for absorption into the bloodstream. The primary digestive enzyme produced in the stomach is pepsin, an enzyme that breaks down proteins into amino acids. The stomach also secretes lipases that break down lipids. The stomach is maintained at a low pH through the secretion of hydrochloric acid by specific cells lining the stomach. Several enzymes have evolved to act at a very low pH of 1 to 3 units. When food arrives in the stomach and digestion there begins, cells lining the stomach secrete a peptide hormone called gastrin. Gastrin secreted by the stomach is absorbed within the small intestine and recirculated to the cells lining the stomach wall. There, the recirculated gastrin stimulates the stomach lining cells to increase their production of gastrin, forming a positive feedback loop. The feedback loop ensures that protein digestion occurs in response to the start of a meal as food enters the stomach. If the pH of the stomach becomes too low, gastrin secretion is inhibited by negative feedback control. Several mechanisms protect the stomach itself from the acidic environment and the presence of digestive enzymes. Glands in the lining of the stomach secrete mucus to protect the stomach wall. Digestive enzymes are also secreted in an inactive form, otherwise the stomach cells would be digesting themselves. After secretion, the inactive enzymes are activated by a change in their chemical structure. The stomach walls contract to mix the content of the stomach, aiding their digestion. Waves of muscular contraction called peristalsis move the food toward the base of the stomach. There, the pyloric sphincter, a band of muscle, opens and allows small amounts of digested food to enter the small intestine. Opening and closing of the pyloric sphincter regulates the rate at which the stomach empties, allowing time for digestion and absorption of the food products released into the small intestine. Animals often eat large amounts of food in a short time, but it takes much longer to digest that food. Therefore, the stomach serves as a storage compartment as well as a digestion compartment. In humans, it typically takes about 4 hours for the stomach to empty, allowing digestion and absorption of nutrients to occur between meals. In other animals, stomach emptying can take much longer. Most carnivorous animals consume large and infrequent meals, resulting in long periods of digestion and nutrient absorption compared with humans. Snakes, such as pythons that engulf large prey whole, elevate their metabolic rate to high levels during digestion, and spend several days digesting and absorbing the nutrients from their meal. This requires an extensive remodeling of their gut to produce new secretory and absorptive cell surfaces to digest their single large meal. These extra cells are not retained between meals because of the high energy cost of maintaining them. After the food enters the small intestine, final digestion of proteins, carbohydrates, and fats occurs. The nutrients released by digestion are then absorbed by the body. The small intestine is made up of 3 sections. The initial section is the duodenum, into which the food enters from the stomach. Most of the remaining digestion takes place in the duodenum. Nutrient absorption takes place in the duodenum as well as in the next 2 sections, the jejunum and the ileum. 2 accessory organs, the liver and pancreas, aid in the digestion of proteins, carbohydrates, and fats in the duodenum. The liver produces bile, which aids in fat digestion by breaking large clusters of fats into smaller lipid droplets, a process called emulsification. Bile allows lipases to digest the lipids more effectively. Bile produced by the liver is stored in the gallbladder. When fats enter the duodenum, cells lining the duodenum release a peptide hormone called cholecystokinin, which causes the gallbladder to contract, thus releasing the bile into the duodenum. The pancreas functions as both an endocrine gland secreting hormones directly into the blood and an exocrine gland secreting substances into ducts that connect to the duodenum. The pancreas produces a variety of digestive enzymes, including lipase, which breaks down fats, and trypsin, which further breaks down proteins. Like pepsin produced by the stomach, the pancreas produces trypsin in an inactive form called trypsinogen to avoid digesting itself. Finally, stomach acid in the duodenum stimulates cells lining the duodenum to produce secretin, which stimulates the pancreas to secrete bicarbonate ions. These ions neutralize the acid produced by the stomach to create an environment for the intestine enzymes to act. Enzymes that break down proteins work best in acidic environments like that of the stomach, but most proteins, like those that break down carbohydrates and fats, are denatured in acidic environments. When large macromolecules are broken down by digestive enzymes, they are ready to be absorbed, another function of the small intestine. The vast majority of absorption occurs in the jejunum and ileum portions. These 2 sections of the small intestine have highly folded inner surfaces called villi, and the cells constituting the villi themselves have highly folded surfaces called microvilli. Together, villi and microvilli greatly increase the surface area for the absorption of nutrients. Cells that line the small intestine are connected by tight junctions. These junctions force the products of digestion to be absorbed across the microvilli surfaces, controlling the movement through the cell and into the bloodstream, rather than linking between cells. The microvilli of the cells lining the small intestine also secrete enzymes that cleave peptides into amino acids, which can then be absorbed across the plasma membrane. These microvilli also secrete enzymes that break sugars into their subunits, which can also be readily absorbed. Nutrient molecules such as glucose and amino acids are often co-transported into cells lining the intestine with sodium ions from gut contents. The sodium ion binds to a transmembrane protein on the surface of cells lining the intestine that also binds the nutrient molecule. Absorption of the nutrient molecule into the cell is driven by the movement of the sodium ion down its concentration gradient. Because the products of fat digestion, fatty acids and glycerol, are lipid soluble, they do not require a carrier protein for transport. Instead, they readily diffuse across the lipid plasma membrane, entering the mucosal cell, where they are further broken down and transported into the bloodstream. After the absorption of nutrients in the small intestine, water and mineral ions are reabsorbed in the large intestine, or colon, of the hindgut. By the time the gut contents reach the large intestine, the nutrients have been absorbed into the body, but water and inorganic ions remain. These compounds are absorbed in the large intestine until the contents form semi-solid feces. Excessive water absorption can cause constipation, whereas too little water reabsorption results in diarrhea. Diarrhea and constipation also can be caused by toxins released by microorganisms, such as the bacterium Vibrio cholera, the cause of cholera, in the food an animal eats. Digestion is not a solo endeavor. Large populations of bacteria reside in the small and large intestine and help extract nutrients that the animal's body cannot extract itself. The principal gut resident is Escherichia coli, or E. coli. The bacteria nourish themselves by aiding in the digestion of the host's gut contents. They also provide nutrients in certain vitamins, such as biotin and vitamin K, that the animal cannot produce itself. Vitamin deficiency can sometimes result from prolonged antibiotic medication that kills large numbers of gut bacteria. The mutual benefits to the host animal and the bacteria ensure the success of this symbiotic relationship. As we've been discussing, the digestive tract is not a passive tube, but rather secretes enzymes and other chemical compounds, absorbs nutrients, and actively moves food through the body. The digestive tract is made up of several layers of tissue, each with a specialized function. The central space, or lumen, through which the gut contents travel, is surrounded by an inner tissue layer called the mucosa, which has secretory and absorptive functions. Surrounding the mucosa is the submucosa, a layer containing blood vessels, lymph vessels, and nerves. Outside these layers are two smooth muscle layers, each contracting in a different axis. An inner, circular muscle layer contracts to reduce the size of the lumen. An outer, longitudinal muscle layer contracts to shorten small sections of the gut. These two muscle layers contract alternately to mix gut contents and to move the contents along the digestive tract from compartment to compartment. Between the two muscle layers are autonomic nerves that control the contractions of the two sets of smooth muscle. An outer layer of cells and connective tissue, called the serosa, covers and protects the gut. The gut is supported in the abdominal cavity by a membrane called the mesentery, through which blood vessels, nerves, and lymph travel to supply the gut. Herbivorous animals evolved specialized digestive tracts that enhance their ability to digest cellulose and other plant compounds. Most herbivores, such as cows and termites, lack cellulase, the enzyme that breaks down cellulose, a primary component in plants. Instead, they have specialized compartments in their digestive tract that contain large bacterial populations, which do produce cellulase. This association of herbivores and their gut microbes is another example of a symbiotic relationship. Rather than the single stomach of other mammals, ruminants, which include cattle, sheep, and goats, have a four-chambered stomach that is highly specialized to enhance the ability of their gut bacteria to digest plants. The first two chambers, the rumen and the reticulum, harbor large populations of anaerobic bacteria that break down cellulose by fermentation. Some of the products are used as nutrients by the host. Carbon dioxide and methane gas are also produced as a result of bacterial fermentation. In contrast to the foregut fermentation of cows and sheep, other mammalian herbivores, such as koalas, rabbits, and horses, digest the plant material they eat by hindgut fermentation. Hindgut fermentation occurs in the colon and in the cecum, a chamber that branches off the large intestine. Because the fermentation products released from the cecum of a koala or horse have already passed through the small intestine, which is the main site of nutrient absorption, hindgut fermentation is less efficient than foregut fermentation in terms of nutrient extraction. In humans and other animals such as lemurs and squirrels, the cecum is present but small, and it includes the appendix, a narrow, tube-like structure that extends from the cecum. The appendix is an example of a vestigial structure, a structure that has lost its original function over time and is now much reduced in size. This brings to a close our discussion of animal metabolism, nutrition, and digestion.